Hello everyone, how are you? So we will start the lecture 7. I would like to remind you that we will have the midterm exam next Tuesday, and I would like to remind you. Can you hear me? It's okay. I would like to remind everyone that we will have a no question, zero question policy during the exam. And the reason is because some students take the exam here, some students take it in the Triton testing center because of accommodations. And so we want to make sure we are fair with all students, even students that take the exam within the classroom. Not everyone feels comfortable asking questions. And so the exam is checked and double checked and triple checked, and so there should not be any errors. However, you will have a space at the end of the exam to raise any concern you may have in a particular question that you think is incorrect or erroneous, or and we will consider that. And if you are right and there is a mistake with the question, then that will be applied to all exams. I think that this is the most, after many years, I think this is the most fair way to go. And so it will be a zero question. You need to do the best you can do. And so TAs and myself will be proctoring the exam, but will not be answering questions, okay? That will be added to a version in the syllabus, so it's very clear. And I will also send an announcement. Okay. Okay, so today we will learn more about B-cell development, and I hope that you have been able to do that with the pre-recorded lecture. And so this is a very short lecture, intentionally, so there is also time to answer any question you may have from previous material that will be included in the exam. And so, to make sure that all your questions are answered. I also remind you that I have office hours today, and so you can bring your question for a small group discussion, whatever you feel more comfortable with. The goal here is to provide you with every opportunity for you to answer all your questions before the midterm, okay? And so you do great. Okay, so this should be working now, and we should be able to start. Okay, so as always, we will start with the summary. And so we learn more at the molecular levels, how the BCR is in germline and how it is rearranged, and also how splicing may occur, and ultimately to have a functional BCR. And so that occurs during B-cell development. So you already know that B-cells develop in the bone marrow. That's why they are called B-cells, because they develop in the bone marrow. And they derive from this undifferentiated common lymphoid progenitor, right? And so from the common lymphoid progenitors, then there are different stages in development to generate these naive B-cells that we were talking about, that were the ones that will encounter the antigen and will be clonally expanding. So what we will learn now is what is going on, what are the stages on development, and how this matches with different rearrangement of immunoglobulins during this gap that from the common lymphoid progenitor into the naive B-cell that is circulating, and that we learn that when see antigen will expand, correct? So the goal of this the B-cell development, what is the goal? There are two goals, right, that we need to, the B-cell needs to accomplish. when it develops and they make sense. So what are the two goals? Yes. One specificity. So each B cell will have identical, at a given time, will have identical BCRs. And that means that we'll have only one specificity, right? And the neighboring B cell will have identical BCRs in their surface and will have another, may have most likely another specificity for antigen, okay? So that's one goal, that each B cell has one and only one specificity. And the second is which one? Yes. No self-reactivity. And so that also makes sense. And it's not perfect, but we eliminate during development many B cells specific for antigens, particularly those that reach the bone marrow, right, where the B cells develop. And so that's exactly right. We need that to avoid autoimmunity, okay? So let's see how is that accomplished. So the common lymphoid progenitor will give rise to the, oops, we have a lot of math here. Will give rise to the, which is the next one, that's the first stage of B cell development. Early pro-B cell, then we have late pro-B cell, then we have large pre-B cells and small pre-B cell, and then we have the immature B cell, and after that, we have the mature B cell, that again is the one that expresses IgM and IgD, that is the one that will circulate, that we have been talking about, okay, the naive B cell. Okay, so the first thing that occurs, and since I cannot write there, maybe I can, yeah, okay. So the first thing that occurs is the rearrangement in the heavy chain loci, okay? And so we have two copies of the heavy chain loci. We will start rearranging one copy, and the DJ will be the first one, okay? And the B and one D and one J segment will come together, and then it will come together with a V. And so ultimately you have a VDJ that will encode the variable region of the heavy chain that will be transcribed with C mu and C delta, but in this case only C mu will be expressed, okay? And so this C mu is the heavy change for the immunoglobulin, right? At this stage of development, which now we are in a late pro-B cell and large pre-B cell, when the mu change is expressed is fully rearranged and is expressed in the surface. At this stage of development, the mu change is expressed in the surface with a surrogate light change, a placeholder, because at this stage of development, the variable region of the light change has not yet been rearranged, okay? Makes sense? Any question about that? And so this mu change with the surrogate light change is form what is the pre-BCR, okay? It's a pre-BCR that has the heavy change, two identical heavy mu heavy change, and then two fake or surrogate light chains. These surrogate light chains are constituted by two polypeptides that are B pre-B and lambda 5, and then these are polypeptides that are encoded in germline, okay? There's no rearrangement. Okay, so once this pre-BCR is expressed in the large pre-B cells, so now this pre-BCR will help the B cell get the first checkpoint, first checkpoint. That basically, like all, the second checkpoint as well, has two outcomes, right? The first outcome is what we call allelic exclusion. And basically, any pre-B cell expressing a pre-BCR that is functional means that was able to rearrange a functional polypeptide that is as the heavy change of the immunoglobulin, will have a induce inhibition of the RAG proteins and other mechanisms that will inhibit subsequent rearrangements in the immunoglobulin heavy change in the other chromosome, okay? And so that's allelic exclusion. And what is the goal for that? Why that makes sense? Or what will be the ultimate consequence of the allelic exclusion? Yes?
The goal of that is to have one specificity.
Exactly. Because now, if we allow rearrangement in the second chromosome, in the second copy of the immunoglobulin heavy change, now this rearrangement will be random, right? So it may give another polypeptide and then we will have two functional polypeptides potentially that have different specificity in one B cells. And we don't want that. We want just one specificity per B cell. Okay. So the second consequence of passing this checkpoint as a the first checkpoint as in a pre-BCR is proliferation, and the third one is differentiation. And why this makes sense? What does it mean that we passed the first checkpoint? Yes. Exactly. That the rearrangement, which can go wrong for so many reasons, because remember there are nucleotides that are added and extinct, and so it can go wrong. But if the pre-BCR is in the surface, it means that the rearrangement in the immunoglobulin heavy change went well, okay? And that is something we want to capitalize on. And so that's why we make copies of this B cell that was successful rearranging the immunoglobulin, and so that's the proliferation. And then we allow differentiation, so now in the next step of development, which is the small pre-B cell, the light change can start being rearranged, okay? And so the light chains start being rearranged after the first checkpoint of the pre-BCR is passed, okay? So if that is not passed in the let's say that the first copy of the immunoglobulin heavy change is rearranged unsuccessfully, and so that will not pass the first checkpoint, and so the second, and so there will not be allelic exclusion. So the RAG genes will continue being expressed, and that will induce rearrangement in the second copy of the immunoglobulin heavy change, okay? And so there are two chances of rearranging a functional heavy change. First, one copy in the chromosome, if successful, that inhibits the second copy to be rearranged, but if that's not successful, the second copy can be rearranged. And so we have two chances of generating a functional heavy change that will be tested in the form of a pre-BCR through this first checkpoint, okay? Any question about that? Yes? It will only add one because you have only one more copy of the immunoglobulin heavy change in the other chromosome. Yeah. Any other question? Okay, so let's say that if it doesn't pass, if it's not able to rearrange a functional heavy change, then this cell will die, okay? And actually, we know that many cells die in the bone marrow during B cell development, okay? So now, if it is successful, past the first checkpoint with a pre-BCR, now it will become a small pre-B cell, start rearranging the V and J for the immunoglobulin light chain, and form now a BCR in the surface. If that rearrangement is successful, we already had at that point a functional mu chain, right? A functional immunoglobulin heavy chain, correct? Because that's why it passed the first checkpoint. So now, with this BCR, this mu chain will be accomplished with the real light change that was just rearranged, correct? And that's why this is a BCR. And that's expressed in immature B cells, okay? And that's it's an immunoglobulin M BCR that is expressed in the mature B cell. And that will be tested in the second checkpoint. And the consequences are exactly the same. So if the B cell, if the mature B cell passed the second checkpoint, means that it has a functional BCR, a functional immunoglobulin M expressed in its surface, now the consequences are the same. Allelic exclusion, and so it means that it will inhibit any rearrangement in the immunoglobulin light chain. Proliferation, because we want to capitalize on this B cell that already rearranged well the heavy and the light chain. And differentiation, so now we can generate the naive B cell or mature B cells that we have been talking about that will express IgM and IgD in their surface, okay? If the reasoning is exactly the same with the second checkpoint. If the immature B cell or is not or the small pre-B cell is not rearranging a functional BCR, when it start rearranging there are 4 copies of the light change. 2 for the kappa that have the kappa constant region and 2 that have the lambda constant region, okay? We said that it's not important or we don't know any biological difference between or effector function difference if you have kappa and lambda, but it's helpful to have 4 copies of immunoglobulin heavy chain genes, because now we'll start arranging the kappa in the first chromosome, and if that rearranged successfully, now it will pass the second checkpoint. If that is not successful, now we start arranging the second copy of the kappa, and if that's successful, it passed the second checkpoint. If that's not successful, now we have a third chance. Now we can rearrange the first copy of the lambda constant of the immunoglobulin light chain that has the lambda constant region, and then if not successful, then the second copy of the lambda constant region of the immunoglobulin light chain that has the lambda constant region. And so that means that gives 4 chances. We have 4 chances to rearrange successfully the immunoglobulin light chain, okay? Because just because there are 4 copies of these gene loci, okay? Okay, so any question about that, the different checkpoints, and I'm happy to answer any question that you might have.
I was just wondering, whenever the B cell fails the checkpoint and dies,
Yeah.
should the signal be given by squamous cell as well?
Oh, that's a very interesting question. Okay. When the B cell express the pre-BCR, I'll answer your question. When the B cell express the pre-BCR or the BCR and is functional, what is very interesting is that there's no known antigen or ligand to that. It's just the expression of these proteins in the surface that induce some signaling that drives the survival and, again, allelic exclusion, proliferation, and differentiation, right? So now, if this pre-BCR or BCR are not expressed because there was no functional rearrangement, then this B cell will die. I think I'm sure the death is via apoptosis, which means that it's a non-inflammatory type of death. It's like a homeostatic, I would say, type of death, and so it will not create inflammation. It's a healthy type of death, basically. I think that the default is that the B cell will go into apoptosis unless it expresses the pre-BCR or BCR. So there's no need for something inducing that apoptosis, right? So that's how I understand it. Yeah. And what is interesting, and it's true for T cells as well, is that there are many phagocytic cells that will be cleaning up these apoptotic B cells or T cells that will die during development because they didn't rearrange in functional BCR, and we will see for T cells is the same with the functional TCR. Mm-hmm. Okay, any other question? Okay, so we already talked about the sequence of the immunoglobulin rearrangement. First is the heavy change DJ, then the heavy change BDJ, and then last the light change BJ segments that are rearranged. We already talked about allelic exclusion. We said that the functional pre-BCR or BCR, when they are expressed without the need for any ligand, will send signals to stop further rearrangement and also proliferate and differentiate. But this stopping of the further rearrangements is what we call allelic exclusion, and it will lead to accomplishing the goal of having just one specificity for each B cell. Okay, so then we have the other goal in B cell development is not to generate B cells that are self-reactive, correct? And so that's occurring through negative selection and receptor editing. So negative selection is basically a mechanism via which that occurs in immature B cells. So once the BCR is formed, the second checkpoint has passed, and these immature B cells will, if their BCR is functional, right, because it passed the second checkpoint, if their BCR is receiving seeing its ligand, its antigen in the bone marrow, that will induce a strong signaling inside the B cell and death of this immature B cell. And so this means that any B cell that has a specificity for antigens that are in the bone marrow will die, okay? And so that will eliminate many self-reactive B cells, and we will learn later that it's not perfect because there are antigens that are not in the bone marrow, right? And that means that there are some B cells that are self-antigens, for example, insulin or antigens that are in the eye, and so there are tissue-specific antigens, right? And so these ones are not in the bone marrow, And so the B cells that are specific for these antigens will not be deleted, and it will leave the bone marrow. And that's why we do have some B cells that are self-reactive, okay? And those are the B cells that at some point may cause autoimmunity. However, we will learn that there are other mechanisms via which we can suppress these B cells or prevent them to be fully activated and secreting high affinity antibodies that can cause autoimmunity. Maybe you can guess what is one mechanism, because the B cells need help from T cells, right? From most parts, right? And so there are mechanisms to eliminate T cells that recognize tissue-specific antigens during the development. And so we will not have these B cells that are not deleted in the bone marrow will not have help and will be, will not be able to generate high affinity antibodies. And that's why most of us do not develop autoimmunity, correct? But they are a potential risk. And so if everything is not working perfectly and something happened with T cell, for example, development, and so you might have autoimmunity. And so we will learn that which are the things that may work wrong to cause autoimmune disease, okay? Sounds good. But all the B cells that recognize antigens in the bone marrow, self-antigens in the bone marrow, will die due to negative selection, okay? Now, there are some chances of an immature B cell that may recognize the self-antigen, and it would be destined to die, but it has other chances to rearrange immunoglobulin light change loci. And we said that we have 4 copies of that, and so depending on how many have been used, it can continue rearranging the leftover copies in a way that maybe that rearrangement, additional rearrangement, additional opportunity to rearrange may change the specificity, because the heavy change cannot be rearranged anymore. But by rearranging the light change and generating another light change with another variable region, that, because the antigen binding site is formed by both the heavy and the light change variable region, that will change the antigen binding site, right? If you put a new light change, and that will lead to the changes in specificity, okay? Any question about that? Yes. Sorry, just to say, these additional chances for rearranging the immunoglobulin light change, if the B cell is self-reactive, are called receptor editing. It's a process called receptor editing that actually was discovered by a scientist here at the Scripps Research Institute. And if it's still self-reactive and there's no more copy of the immunoglobulin light change to be rearranged, then the cell will die. Okay, yes. 2 questions.
So that was pretty much my question. So it's an order, it tries to rearrange the light chain first, and then if it's unsuccessful.
Exactly. So it starts rearranged first, the first copy of the immunoglobulin light chain that contains kappa, encodes kappa, then the second copy that encodes kappa, and then the first that encodes lambda, and then the second that encodes lambda. And basically can use all of them to pass the second checkpoint or to avoid negative selection. And then when there's no more, then and if that has not been successful to either pass the second checkpoint or to avoid negative selection, the cell will die.
After the second chance of heavy chain rearrangement, does that also change specificity?
Good question. So we talk about, now we are talking about at the stage of pre-B cell, there will be the pre-BCR expressed, and that will be the first checkpoint. And so if the first copy of the heavy chain has not been rearranged successfully, there is a chance to arrange the second copy in the other chromosome. If that's successful, there will be a new pre-BCR that is expressed, right? And so if the question is if that changed a specificity. Anytime you rearrange a new BDJ, right, like that will be random, remember? And so that will be a different specificity, right? However, in this case, it's a little bit tricky because the reason why the second copy of the heavy chain is rearranged is because the first one didn't rearrange any functional polypeptide, so there wasn't any specificity there, right? And so that's what, yeah, so it's tricky, the question of whether there will be a change in specificity because there wasn't any functional pre-BCR to begin with. Okay, but you remember the BDJ basically is changing the sequence of amino acids in the variable region. Anytime you change the sequence of amino acids in the variable region, the antigen specificity most likely will change, right? Because the chances that you rearrange exactly the same, put exactly the same nucleotides, excise exactly the same, are very low or zero. Okay. Any other question? Last, we learned about the X-linked amyoglobulinemia. This is an immunodeficiency that occurs in individuals that have mutation, loss of function mutation in this kinase that is called Bruton tyrosine kinase, and that is encoded in the X chromosome. So that's why mostly affects males because they have only one copy of the X chromosomes. Females have two, and so they may have still a wild-type copy of BTK. And basically, this BTK is a kinase that is important for the signaling of the BCR. And remember, we mentioned in the pre-recorded lecture that the BCR has a very short transmembrane region. And so it's always accomplished with the immunoglobulin beta and immunoglobulin alpha. I'm sorry that, there are many names here, but these are just polypeptides that are like any other protein, most of the proteins in our body that are encoded in the germline, right? And so there's no rearrangement for that. These are short chains. But what they do is facilitate the signaling because they have a longer transmembrane region, right? So they are kind of addressing this limitation of the BCR. And so this associates with BTK and it allows the signaling. And so we said that for passing the first checkpoint and the second checkpoint, you need signaling through pre-BCR and BCR respectively, right? Even without the ligand, but you need the signaling. And so if you don't have BTK, where the cells will arrest in development? Will they pass the first checkpoint? No, right? And so they will not generate ever immature B cells, okay? Or small pre-B cells, because they will not pass the first checkpoint. And so basically these individuals have low immunoglobulin levels, enhanced infection, particularly with some encapsulated extracellular bacteria, which because of this capsule that they have, and I don't know, I'm not a microbiologist, but what my understanding is these bacteria evolve a capsule that make phagocytic cells like neutrophils not see them or not uptake them. And so for them to be uptaken and be able to be degraded by the phagocytic cells like neutrophils, they need to have an antibody, particularly immunoglobulin G2, recognizing antigens in these capsules, and now through Fc receptor or fixation of complement can opsonize these encapsulated bacteria and now can kill them through neutrophil phagocytosis. And so if there's no B cells, there will be many infections, particularly by these bacteria that require antibodies to be eliminated. Any other question? Yes.
This is not really specific to this topic, but I was
Yeah.
wondering, like, in terms of the bigger picture, is it better for us to have as much antibody to recognize as much pathogen as possible, or is it better for us to avoid autoimmunity?
It's a trade-off, right? I think that my thought is that we need to defend against pathogens because otherwise, many immunodeficiencies are lethal, right? And so clearly there is a need, and they are lethal in early age. Autoimmunity are chronic diseases that not necessarily are lethal. And so, I think that it takes priority to generate billions of specificity so you can survive a world full of microbes, right? Excellent question. Okay, shall we do a clicker then? Okay, so can you connect? I don't see connection in the base. Yes? Can no, right? Okay, so let me stop it and you continue with this and I will start this clicker again. We will do this one again, okay? You will have to vote again. Now it works. Yeah, it's the order in which I start things. Okay, so please vote. Everyone vote again to make sure that you get the points because I had to restart the class. Okay, so make sure everyone voted, and then we will discuss this one. Okay, I'll stop it now. And so which of the following cells can be found in the bone marrow if the productive light chain rearrangement does not occur? Okay, so the productive light chain rearrangement will be necessary to form a BCR, right, in an immature B cell. And so the immature B cell will not be there because there's no functional BCR, right? But you will have pro B cells you will have pre-B cells because there was the pre-BCR checkpoint has passed, and there will not be immature B cells, again, because these immature B cells are the ones that have the functional BCR, which will not be expressed if immunoglobulins are not present, okay? Okay, so let's, most of you answered the correct one, which is D. And so, any question about that? Yes.
So just to clarify,
Yes.
when the pre-BCR is expressed and when the BCR is expressed, that means that it's functional, right? There's not like a case where it's expressed but not functional.
Oh, excellent. Absolutely. Exactly. When it's expressed, it's functional. Yes, and it will induce this signaling that is necessary for these three outcomes: allelic exclusion, proliferation, differentiation. Yes. Okay, so let's do the next one. Okay, I'm going to stop it now. And there are between D and E, and so I want you to talk to each other, to your neighbors, and then basically think if you will be able to rearrange the light change locus if you have a B cell or a organism that is defective in lambda 5 gene, right? Will you ever get to rearrange this B cell will ever be able to arrange the light change locus? Okay, so why don't you discuss, and I'll start this poll again. And that's very nice. Yeah. You can discuss with the ones in the back, with the neighbors in the back. What do you guys think? So which one did you pick? C? Inability to produce immature B cells. Yes, that's definitely correct, right? Because if you don't have the lambda 5, right, you will not have a pre-BCR, so there is no passing of the first checkpoint, so you never get to immature B cells. So that's correct. So now what about the A and B? So maybe you guys can discuss A and B or not, if you wish. If you wish, up to you. Okay. Okay, it has polarized a little bit more, and so still, I don't know, maybe I'm wrong. So let's discuss together. So I'm going to stop it now, okay? And so, okay, so most of you answered D. And then some of you answer E and very few the rest. So maybe, whoever answered D, which are the two of the above that were right? A and B? A and C. So A, everyone agree that A is correct? So if it doesn't pass the first checkpoint, so there's no lambda 5, there's no pre-BCR, it will not pass the first checkpoint, correct? And so it will never get to rearrange the immunoglobulin light chain. So that's correct. There will be inability to produce functional light chains, okay? B says that there will be inability to produce a pre-B cell receptor, and that's correct, right? Because the pre-BCR contains a heavy change that is fully rearranged, but the heavy change cannot be expressed in the surface. The pre-BCR needs to contain this surrogate light change to be expressed in the surface. So that's the pre-BCR. It's a BCR, except that instead of a real light change, has the surrogate light change that contain this lambda 5 as an equivalent of the constant region of the light change, and the pre-alpha. Pre-alpha? Am I saying it right? Now I'm blanking on the name. How is it, Joe? What is the name of the other polypeptide in the surrogate light change? B-pre-B, variable, yeah, and that's the surrogate of the variable region. But these are germline encoded polypeptides. But basically they are necessary because the mu, even if fully correctly rearranged, requires the surrogate light change to be expressed, okay, and form the pre-BCR. So there will be an inability to produce a pre-BCR, okay? And so that's correct as well. And C says inability to produce immature B cell, which is true because there will be an arrest in development because the cell cannot pass the first checkpoint. And so the correct answer is all of E, all of the above. Okay, any question? Yes?
I just want to clarify.
Yes.
The lambda 5 gene, that's different from the lambda gene in the light chain rearrangement, right? Or is it the same?
Yes, excellent question. And I know it can be very confusing, but I didn't give the names to this. But yes, it's very different because that's lambda 5 and pre-B, I'm missing again, pre-BV, no, that doesn't sound right. It's lambda 5 and pre-TB. Job, help me out. Let's see, I just need to go to Canvas to this. Sorry, you pass the 50s and B-pre-B. Yeah, you were right. So anyway, so the lambda 5 and B-pre-B, no, but this is the part that I really think that you should remember. These are polypeptides that are encoded in germline. There's no rearrangement or anything. These are fixed, are always the same in every cell. Your hepatocytes also have the same DNA for this. They just don't express it, right? But these are absolutely germline encoded proteins. They are placeholders, okay? And it's very different from the immunoglobulin light change, which has two versions. One version that has BJ with the kappa loci, the kappa as the constant region, and one version that has all the BJ with the lambda as constant region, right? This immunoglobulin loci, light change loci, will be rearranged, needs to be rearranged in the variable region with the V and the J, one B and one J brought together to form a VJ that encodes the variable region of the light change together with either the kappa or the lambda, depending with which loci we are rearranging. Okay? Thank you so much. This was very important to clarify. Thank you. Okay. Any question? So this is the only one that you have answered wrong. Most of the class have answered wrong this question since we started. Yeah.
We've been having like a hard time like compartmentalizing like lambda 5 for the context of like producing like B cells, and then one where it's when it's in the surrogate. Because you're saying that it's two different.
Okay. One thing is lambda 5 that is very different than lambda. Okay? Okay, two different things. Okay? They are just, the names are similar. But lambda 5 is very different than lambda. Is that clear? Yeah. For everyone? Thank you for clarifying. For asking, yeah. Okay. So let's do the next one. Okay, so I'm going to stop it now. And this one, all of you answer correctly. What is true about a patient with X-linked agamaglobulinemia, which is basically with a loss of function mutation in Bruton tyrosine kinase, which is important for signaling of the pre-BCR or BCR, right? And so because there's no signaling in the pre-BCR, it will not pass the first checkpoint. And so there will be lack of immature B cells, there will be lack of plasma cells, there will be susceptibility to extracellular bacteria, we just discussed that, and very low levels of immunoglobulins in the serum. And I imagine it says low levels because it might be, not absolute. I don't understand why there is no zero immunoglobulin in the serum. Perhaps there is still some residual signaling that is very, very, very low efficient that might lead to some B cells to make it through. Biology typically is not black and white, and so I think that it might be related to that. Okay, any question? All of you answered the correct one, which is E. And so I would like to, we can end early, but before I'd like to give the opportunity for you to ask any question you might have for these lectures 1 to 7 that will be included in the midterm. How do you feel about the flipped classroom format? Good? Yes? Okay. Okay, I'm glad that that is being helpful to you. I definitely enjoy it, because it allows more interaction and yeah, it's more fun. And I do believe that you get double for the same tuition. Okay, so I think that, if there are no more questions, you have some 20 minutes extra to study for the midterm, and I'll see you everyone on Tuesday. Take care. and I will have office hours at 10:30. Sorry, 9:30. 9:30.
I do ask a question about from the last lecture. I'm sorry, I cut
Yes.
my tooth, so I couldn't make it.
Yes.
I was wondering, so for the somatic recombination that gives the antibody diversity,